G'day Junior Tuckies, and welcome to this explanation of the Paper 2 for Life Sciences. I'm going to be taking through exactly what you can expect in your Paper 2, and hopefully this will help you better prepare for your final exams at the end of the day. So looking at the slides over here, we're going to look at what makes up your Paper 2. So we're going to be looking at 4 sections specifically, and what constitutes these 4 sections in your preparation. So looking at the term 1 work that you would have already gone through, we're going to be looking at DNA code for life, followed by meiosis. These are very related to one another, so it makes sense that we're going to have them in the same paper. We're going to move on to genetics and inheritance, and then finally we'll be looking at what is expected of you in your understanding for the sections on evolution. Please make sure that when you are preparing for your final exam and studying the work that you see here, as well as the work in Paper 1, you have a copy of the most recent 2021 examination guideline. You're going to find lots of hints, lots of focus points in the examination guideline, so make sure that you have this with you. That's a non-negotiable. Okay? Just looking at how your paper is going to be set up. Firstly, we're going to look at a Section A. Section A is usually your shorter type questions, your level A and B in terms of cognitive expectation, and this is going to make up 1/3 of your paper. Here you're looking at 50 marks. Generally for Section A, we look to get approximately 45 out of 50, at the very least 40 out of 50, especially if you're trying to get a very, very good grade for the papers. Section B, we're also looking at a variety in terms of types of questions, and we're going to have 2 main questions, obviously subdivided, but each of these questions is going to total 50, and then the total of our paper here is going to be 150. All right, so moving on from here, skills in both Paper 1 and Paper 2 is extremely important. Okay, so when we're focusing on skills, we'll need to know how to do biological drawings. We'll need to be drawing graphs, the various types of graphs. So I would definitely say go and revise these. You will definitely get a graph to draw in your final exam, whether Paper 1 or Paper 2, or both. We're going to have a look at our variables and how they're controlled in the context of an investigation, being able to identify the independent variable, the dependent variable, and then focusing on a few of the controlled variables that could impact on the results of an investigation, and then knowing the difference between validity and reliability in the investigation as well. Validity being focused on the variables that need to be fixed so as not to impact on the results, and reliability looking at, well, in this investigation, are we going to repeat the investigation to make sure that our results are reliable, and then also looking to increase a sample size when it comes to the investigation itself. Now, one of the biggest mistakes that students make when answering a question on validity and reliability is getting the two confused with one another, but most importantly, it's actually not applying the concept of reliability to the investigation that you've been given in the question. So I would say, read your question carefully, have a look at what the scientists have done in the investigation to, 1, improve the validity of that investigation, and secondly, what they have done to make sure that the investigation is reliable as well. Okay? In terms of our skills preparation, graphs are easy marks. Okay, so be sure you know how to draw a graph. This includes line graphs, bar graphs, histograms, and pie charts. All right, we're looking at a general setup like this over here, where our type of graph that we've chosen to draw is correct for 1 mark. Headings or captions for the graph are correct, including both of the variables. And here I'm referring to the independent and the dependent variable. Okay? Scale lines, in other words, calculating a scale line for each of your axes, the x and the y axis, that includes all of the points that need to be plotted in the investigation. All right? In life sciences specifically, other than mathematics, we have to include a scale from 0 all the way to the end of the scale line. We cannot compress our scale line close to the origin. You'll lose marks, particularly at this point, if you do that. The mark for scale lines is the skill in calculating the appropriate scale for each of your axes. Putting in a title for each of the axes, make sure that you include your units. This is also an area where some students do lose marks, particularly when drawing graphs. The units will be given to you in the question, so read carefully for the units that you're working with. Or if you're given a table, my suggestion would be look at what you have in the first line or first row of your table, and those will most likely be the labels that you're going to be using for your x and y axis. Okay, and then there are 2 marks for plotting the points in terms of the coordinates on the graph. All right, moving now into DNA code for life. At this point, I just want to tell you that what you're going to be seeing on the slideshow is a lot of information. And I've included that information to assist you in terms of study notes in preparing for your Paper 2 Life Sciences exam. I'm not going to be focusing on every one of the slides, but that is there for you to prepare adequately for your paper. Okay, so let's get into it. DNA code for life makes up 27 marks of Paper 2. This is a fair number of marks, and they're also pretty easy marks. So generally, Section A focuses over here. We might have 1 or 2 drawings, and that's where I'm going to focus on now. So typically speaking, when we're looking at DNA code for life, we focus on DNA and RNA, and particularly the differences between each of these nucleotides. Now, this is the chemical molecule that is used to build up both a DNA strand as well as RNA strands that obviously fulfill different functions in the body. You're going to see over here that we have a very simple base pairing on a DNA strand showing 2 nucleotides. Now, this is a very, very involved drawing. Your drawings, if you had to do, that you would have to do in your exam, would not be to this extent. You would need to represent a phosphate group in your nucleotide. You would need to represent the pentose sugar, and you would need to represent the nitrogen-containing base. Okay? In the case over here, you can see that we differentiate between each of these 2 bases as purine bases or pyrimidine bases. And you can see that the difference between the 2 is the fact that this is a double ring structure and that one is a single ring structure, but that is not something you need to know for your exams. So you would represent a base simply by drawing a square and then labeling it as either adenine, cytosine, guanine, or thymine. That is the DNA nucleotide on its own, and what you see on the screen, both of them, the base pairing on a DNA strand. Okay. Now, one of the most common mistakes when drawing one of these nucleotides is if you're asked to draw a DNA nucleotide, the biggest difference between DNA and RNA in terms of the nucleotides is that in a DNA nucleotide, my pentose sugar is a deoxyribose sugar. In an RNA nucleotide, this pentose sugar is simply a ribose sugar. There's a second difference is when we look at the bases, DNA bases, we're going to have adenine, cytosine, guanine, and thymine. But if we're looking at an RNA nucleotide, the thymine base is substituted out by the uracil base. So that's one easy way to determine which of the 2 nucleotides you're actually looking at. Okay? Now we're going to be looking at this. Now, differentiating between a nucleotide and a nucleic acid. Okay? Students do struggle with this concept a little bit, so we're going to differentiate between the 2. We've just looked at nucleotides, which are the building blocks of nucleic acids. And the 2 nucleic acids that we're going to be doing over here are going to be DNA and RNA. DNA, we're looking at a double-stranded molecule with a pentose sugar that is deoxyribose, nitrogen base including thymine, and it has a helical structure. If we're looking at the other strand, we'll see that this is an RNA strand. It is single-stranded. Its pentose sugar is simply ribose. The nitrogen bases includes uracil in the place of thymine, and it has a more linear structure. Okay? Most of the time in your Paper 2, you could be asked, or it has been asked in the past, that we compare the two strands. And a very, very important thing to have a look at here is read your question carefully, because sometimes the question refers to a nucleic acid, which is the strand, or the nucleotide, which is the actual molecule. And when we compare a DNA molecule with an RNA molecule, all right, we're looking at either the nucleotide of a DNA with an RNA, or we're looking at the bigger picture. We're looking at the double-stranded DNA helix compared to the single-stranded RNA nucleic acid. All right, staying with DNA code for life, we're looking at the processes that we need to know extremely well. One is looking at DNA replication. This is where the DNA makes an extra copy of itself. And the process in that is extremely important for us. Now, on the next slide, I detail this a little bit, but I'm not going to be going through it step for step. It's there for you to go through. Please make sure that you learn the format in which the different processes take place. Okay, you'll get marks for each one of those. Typically speaking, looking at DNA replication, we're looking at 6 marks. Okay, easy to get marks, but also, if we don't list everything, easy to lose marks as well. Protein synthesis, including transcription and translation, we're going to be going through very quickly as well. Okay, so looking at the process of DNA replication in terms of images, we see that we start with a unreplicated chromosome that now needs to replicate. And in order for that to happen, it has to unwind and unzip in order for us to undergo, or for the process of DNA replication to take place. And what we end up with at the end of the day is a replicated chromosome over there. Common errors with regards to this is that our terminology is not perfect. We refer to a single chromosome as unreplicated. We refer to a replicated chromosome when we find there are now 2 chromatids joined at the centromere. Okay, and this is sometimes where we get confused. To differentiate between the two, we refer to unreplicated and replicated chromosomes in this process. Okay, if we look at this in terms of steps, here are the steps that I've mentioned before. We're going to study these steps point for point. There are 8, maybe 9 possible marks here, of which the examiner is looking for 6. So make sure you get at least 6 of these over here. Okay, let's have a look. Protein synthesis. This is the process where the body produces the valuable proteins responsible for the different functions in the body. Here I'm talking about hemoglobin, I'm talking about enzymes. They're all present in the body because of protein synthesis. And in order for the body to synthesize these proteins on a cellular level, the DNA plays an extremely important role here. So the first step is taking that double helix of DNA. It needs to unwind itself first. It needs to unzip to expose the bases to the nucleoplasm. So in other words, this template needs to be exposed to the nucleoplasm for us, or for the cell, to be able to put together a complementary RNA strand. And in this case, it's a messenger RNA, mRNA strand. And it's going to be a strand that forms complementary to the bases present on the DNA strand. And you can see that represented in the image over there. Okay, this is an extremely important process, and it links very, very closely to translation, which is what happens outside of the nucleus. So be sure to go over this and look particularly at where students make the common mistakes. And one of the most common mistakes when it comes to transcription is that we forget that mRNA is an RNA strand where thymine no longer exists in the sequence but is replaced by uracil. So make sure you put that uracil in. If we're looking at translation, this occurs at a ribosome, and you can see the image on the screen over here depicting the ribosome in the middle. We can see that we've got the mRNA strand that has now left the nucleus through the nuclear membrane, through one of the nuclear pores, attaches to the nearest ribosome, and what happens now is free tRNA nucleotides in the cytoplasm bring their complementary amino acids to the ribosome. So they're hanging around, they're waiting for their complementary codon on that mRNA strand to move through the ribosome before they bring in their amino acid to be attached in a specific sequence to produce the polypeptide chain, or that specific protein that is supposed to be synthesized based on the mRNA strand that has now left the nucleus. Okay, once again, at this point, remembering that uracil replaces thymine when we refer to RNA is very, very important. Many students make the mistake of not including uracil into the base sequence. Also, one of the other points of error commonly made is that when we're looking at this, common questions give us a DNA sequence of bases, or an mRNA sequence of bases, or a tRNA anticodon sequence of bases. And we now need to unpack this puzzle to determine which amino acid needs to be placed where in the sequence of a longer protein chain. So it's exactly that, it is a puzzle. So knowing that my DNA sequence of bases, or my DNA triplet codes, are going to have complementary mRNA codons. These complementary mRNA codons are also going to have complementary tRNA anticodons. And that tRNA anticodon is going to be specific to an amino acid that is brought by that tRNA molecule inside of the cytoplasm. Okay, just looking at meiosis now, this is the next section. The number of marks here we're looking at, 21 marks also. Easy marks to get, but also easy marks to lose if we're not too sure what we're talking about. Okay, so firstly, unreplicated versus replicated chromosomes. And in order for meiosis to take place, there need to be replicated chromosomes. So the process of DNA replication needs to take place first, and this takes place in interphase. Okay, then we have homologous or bivalent chromosomes that cross over in meiosis. And we'll talk about which phase this takes place in a little bit. And that's going to result in our genetically unique chromosomes. And then they are going to further divide in meiosis a little bit later. The terminology here is extremely important. Okay? Phases of meiosis. I know this is very, very small on the screen, but we're not going to focus too much on this image over here. But we are going to be looking at the phases of meiosis in terms of prophase, metaphase, anaphase, and telophase twice. Because meiosis actually undergoes division twice. Knowing each of these phases in meiosis 1 and meiosis 2 and the significance of each of these phases is going to give you success at the end of the day in those 21 marks that you're looking for in your final paper. So looking at prophase 1, this is one of the most important phases in meiosis. Because in prophase 1, along with all of those things that occur, all of those processes that occur, we have the following, which we must never forget with regards to prophase 1. And that is the exchanging of genetic information between the homologous chromosomes. Okay? And that is going to happen as per the previous slide, where we see those bivalent chromosomes crossing over the chiasma and exchanging that genetic material. We'll need to know the corresponding image to make sure that we don't confuse our phases when we have to identify a phase of meiosis by looking at an image. So there you can see an example of what prophase 1 would look like in an image. We're going to look at metaphase 1 over there. Now, I like to look at metaphase. Prophase 1 is pretty easy because we're looking at the first phase. Okay, so there's a couple of things we remember for the first phase. Now, how do we know, looking at a diagram or an image, whether or not we're in metaphase, anaphase, or telophase? In meiosis, metaphase, the M should remind you of middle. This is where the chromosomes come together in the middle. And if we're looking at metaphase 1, our homologous chromosomes have not yet been pulled apart. So in other words, we still see them as pairs. And when they're aligning on the equator in metaphase 1, because that's what M means, middle, metaphase, we can see them as we see them in the image over here. My equatorial line runs between the bivalent chromosomes. Okay? Moving on to anaphase, we can see that with anaphase, my centrioles have moved to the opposite poles of the cell. The spindle fibers have already attached to each of those chromosomal pairs, and now they are being pulled apart. You need to ask yourself this question. When we're identifying anaphase 1, has the centromere been split? In this case, it has not, which means we're still working in meiosis 1 when it comes to anaphase. And we're going to contrast this with what we see in Anaphase 2. All right? There's the image, the corresponding image when it comes to identifying these. So make sure you study these. Common errors in the whole of meiosis include that we're not able to identify the significance of each of these phases. Looking here at telophase, we see a nice image over there that shows us we're moving now towards the end of meiosis 1. We can see that I do have now two separate nuclei, a reforming nucleus surrounding the genetic material, and there is now an invagination that is taking place, okay, or a cleavage. This cell is ready to divide. What we do see in each of these cells now, if we had to go and count each of these chromosomes, is that we're at a point where my daughter cell's number of chromosomes has been halved. So we've gone from 2n to n number of chromosomes already in meiosis. You can see that illustrated in the diagram over there. Each of these daughter cells is also genetically unique because of the crossing over that has taken place in, yes, prophase 1. Right. Moving on to prophase 2. Now we're moving into meiosis 2. This is the final stage, or the final set of stages. PMAT happens again in the division to form the gametes at the end of the day. Okay, so in prophase 2, the daughter cells of meiosis 1 undergo PMAT for the second time. And in prophase 2, our nuclear membrane disappears, centrioles move to the opposite poles again, and now each of my chromosomes that consists of two chromatids joined at a centromere are going to be aligning at the equator with the centromere on that equatorial line. Looking at metaphase 1, the equatorial line was between my two bivalent chromosomes. Now the centromere exists on that equatorial line, because the next phase, moving on from metaphase 2, is that these spindle fibers attach to the centromere on either side, and they're going to be pulled apart, and that's what anaphase is. Anaphase is where the chromosomes, and in this case it's going to be the two chromatids that were joined, are being pulled apart to become, in their own right, two unreplicated chromosomes, which are going to exist in each gamete that is going to be genetically unique. Okay, so we need to know the phases in meiosis. Telophase 2 here, we're going to end up with the invagination and the formation of those gametes, as I said now. One of the common misunderstandings over here is that meiosis does produce 4 genetically unique gametes at the end of this process. So once we've gone through meiosis 1, once we've gone through meiosis 2, we end up with 4 genetically unique gametes. Okay. Things to remember and areas for focus. The importance of meiosis. And the importance here is genetic variation. That means the gametes are each going to be genetically unique at the end of this process. We need to be able to compare meiosis 1 and meiosis 2 when it comes to prophase, metaphase, anaphase, and telophase. So looking at prophase 1 versus prophase 2, how do we differentiate between these? Same for metaphase, same for anaphase, and same for telophase. Comparing meiosis with mitosis, there's a little bit of grade 10 work coming out in our understanding of mitosis, both the similarities and differences in these two processes. What happens with regards to abnormal meiosis, and here I'm talking specifically with regards to non-disjunction, and a very common example used here is Down syndrome. Okay, so studied the Down syndrome, a little bit of terminology with regards to meiosis as well. We're looking at aneuploidy versus polyploidy, trisomy versus monosomy. Okay, and then explaining the occurrence of non-disjunction in anaphase 1 or anaphase 2. And I've included 2 images over there for you to have a look at exactly what happens when non-disjunction takes place. Okay, now here we're looking at 48 marks. Our next section, genetics and inheritance. Okay, a lot of practical application is going to be expected of us in this section, so we need to make sure we know exactly what we are going to be doing. Knowing the necessary terminology and laws with regards to genetics, very, very important. Okay, what's coming out more and more in the exams is knowing Mendels Laws. Now, there are 3 of them, and being able to define each of these is extremely important for us. So the first law, law of dominance, we're going to expand on that just a little bit. Principle of segregation is principle of independent assortment as well. Okay, knowing the difference between a phenotype and a genotype, homozygous versus heterozygous pairing of genes, genetic crosses and pedigree diagrams, which we will go through in a little bit more detail, and then the mutations and paternity testing. All right. If we're looking at the law of dominance, or his law of dominance, we apply his law of dominance to the genetic crosses that we're going to be doing in a little bit. Okay. So, father of genetics. So, when we talk about any of Mendels' laws, something that's come into the examination guidelines that wasn't really an emphasis before is how we refer to the different alleles. Okay, so back in the day when Mendel was doing his studies, alleles were unheard of. So he referred to the alleles for each of the versions of a trait as a factor. So when we are explaining any of his laws, we need to make sure we use the word factor instead of allele. Okay, and this is something new that's come out in the examination guideline very, very recently. Okay, so if we're looking at his law of dominance, okay, I've put it on the screen over there for you. It's very simple. When 2 homozygous organisms with contrasting characteristics, so they're crossed with one another, what you're going to see in the offspring is going to be the dominant trait in the P1 generation. So this basically means when we're looking at a trait, and we know that the parent, the P1 generation, okay, has a combination or has a specific phenotypic trait, and we know it's homozygous. The other parent, we know is homozygous, has a different trait. What we actually see physically in the offspring is going to be the dominant trait in terms of that genetic pairing. Okay, there's an image to illustrate it for you, so spend some time on that image just to make sure you understand exactly what the law of dominance means. Okay. When we're looking at his principle of segregation, it means that, well, we know that every one of the traits within us, within organisms, exists because of the pairing of genes from the parents. So we know there are 2 versions or 2 factors for each of these genes, and they segregate, they separate in the formation of these gametes. Okay, so like we did in meiosis, we know that at the end of the process of meiosis, we have one unique combination of the different factors for each of the traits that we're going to be seeing in the organism. Okay, and we see it over here in meiosis 1 and meiosis 2, as I've just mentioned to you. So where we had a pairing of chromosomes at the beginning in the parent cell, at the end of meiosis in each of the gametes, we only have one unreplicated chromosome of each. His principle of independent assortment refers to how in this process of meiosis over there, no factor is dependent on any other factor when it comes for the sorting in producing those gametes at the end of the day. So there are his 3 laws. Okay, now applying what we've just learned about Mendel to genetic crosses, monohybrid crosses, here we're going to have to understand the interactions between the different alleles. Be sure to practice an example of each one of these in the monohybrid cross context. Okay, looking at complete dominance, this is where we are looking particularly at how one allele completely masks the expression of another allele when it comes to determining a phenotype. An incomplete dominance is where we have 2 alleles where neither of them are dominant over the other, and an intermediate phenotype is expressed, and a codominance is where each of them is equally dominant, and both of those genes are expressed in a phenotype. Okay? Looking at monohybrid cross, this could include, okay, sex determination as well as any of the sex-linked conditions, which we will focus a little bit on later. Okay? Monohybrid crossing in the determining of blood groups, there's a very unique one. This comes out all the time because it's a very good example to use when we try and explain the context of complete dominance and codominance. We can use blood groups for that. Okay? That's why they like to use blood grouping and a genetic cross to explain blood grouping in your final exam. Dihybrid crosses is simply taking that monohybrid cross and we're adding another genetic trait to work out the proportions that each of these are going to be paired in an offspring. Right. So in terms of complete and incomplete dominance, I've already put that in for you. And if you see over here in an incomplete context, we can see that when each of the parents has a specific phenotype, neither of those phenotypes exist in the offspring, but we have the intermediary. In the codominance context, we're going to have both of the phenotypes expressed in the offspring. So spend some time on going through this. It's going to help you prepare for your final exam. Okay, there are also some very nice videos on the Junior Tuckies online platform for you to go and get a better understanding of these. Okay. The format in which we complete a genetic cross is a standard format. Okay, so be sure to practice this, to get to know this, because what we're going to find is that we have to include specific points for marks. Identifying a P1 and an F1 generation gets you a mark. Placing meiosis and fertilization as processes into this genetic cross gets you another mark. So in all intents purposes, out of 6 marks, you've already got 2 marks without even doing the genetic cross. So the first step is to identify the phenotypes of the parents. And in this case, we're looking at sex determination. And we know that the phenotype in this case is going to be that one parent is a male, one parent is a female. And in the genotypes, XY and XX pairing of the 23rd chromosomes. Meiosis takes place and gametes are formed. And each of the gametes in this case are going to either have the X and the Y in the case of the male, and the other gamete, the X and the X in the case of the female. And when fertilization happens, what we're going to have is we complete the Punnett square, and we see that in this case, we have a 50-50 chance of offspring being female or male. Now, how do we apply this to sex-linked disorders? Okay. We're going to be doing the Punnett as you see here, except we're going to include the disorder in both the phenotype and the genotype by including the superscript that you are given in your question for that specific disorder. Now, one of the other things that we need to make sure of, and it's also a common mistake that is made by some students, is whether or not this sex-linked disorder is actually a dominant or a recessive disorder. And we need to apply our understanding of dominance to the context of the question as well. So we're going to complete the genetic cross as you see over there. We're going to include the disorder, and most likely going to be on the X gonosome. So we're going to include our letter on the X, and we're going to filter it through our Punnett square until we get a F1 genotype-phenotype expression. Okay, and don't forget that when we're looking at phenotypes and we're working with a sex-linked disorder, expression of sex is also a phenotype. So we're not only looking, is the genetic condition present in the offspring, but we want to know if it is present within the males or the females of the offspring. Okay, here you can see an image that shows X-linked dominant inheritance in terms of disorders and the X-linked recessive inheritance as well. So spend some time going through this just for a better understanding. Okay. Looking at blood groups, we're going to complete the genetic cross as we do any other cross. But now we need to understand that when it comes to blood groups, we're looking at the interactions of multiple alleles, multiple possible alleles coming together after fertilization. And we know that this results in blood group A, blood group B, or blood group O. One that is not there is the codominance between blood groups A and B. In other words, the allele for the expression of blood group A pairing with the expression or pairing with the allele for blood group B. This gives us blood group AB, an equal expression in the phenotype when it comes to producing blood groups. The allele for blood group O is recessive, which means the only time an individual will have blood group O is if we have the pairing of two recessive alleles. Then we have the expression in terms of blood group O. Okay, and here is a little bit of a reminder of what I've just said to you. So spend some time on that. This is very, very important, and it comes up in every single paper. One of the things that I just want to point your attention to as well is that when we are writing down the symbols for the alleles in terms of the genotype, we need to make sure that we are using the symbol that you see there. If we write down a capital letter I, like you see at the bottom over there, it's no longer going to be accepted. Okay, and this is something that's going to cost us marks unnecessarily, because the concept of determining blood groups we know. Something as simple as how we write down our letter I, costing us marks at the end of the day. We don't want that. Okay, so please reflect back on this and how we put together the symbols to represent each of those alleles. Okay, dihybrid cross, we approach the same way as a monohybrid cross, except we're going to be looking at the pairing of two traits in the offspring. And here I'm going to refer you to the resource videos on the Junior Taki site online, okay, for a more detailed explanation on how to complete a genetic cross. Those videos are fantastic. Okay, I don't want to spend too much time on that today. Pedigree diagrams. You're most likely going to get one of these in your final exam. Yes, you'll have to go and you'll have to work out the different genotypes and potentially the phenotypes of specific individuals within these. And this is how we're going to approach questions like that. So looking at pedigree diagrams, you'll get given an image similar to the one that you see on the right-hand side of your screen now. The first step is to look at the key. Which of these individuals are male, which of these individuals are female? Especially if we're looking at a sex-linked disorder that's represented or that is traced through the generations in our pedigree diagram. Okay? If it's not sex-linked, okay, our job becomes a little bit easier. Because all we have to do to start off with is we identify the expression of a phenotype in the homozygous recessive condition. So here we're going to be looking at the two recessive occurrences, and we see that they occur in females over there. And we know that how we represent the genotypes of each of those individuals is going to be the recessive pairing. All of the other individuals that you see in our pedigree diagram over there obviously do not have the condition. So they're going to either be one of two things. They're going to be homozygous dominant, or they're going to be in the heterozygous condition. And now it is for us to work this out. Looking at this pedigree diagram over here, if the individual that you see at the bottom of the screen over there has a homozygous recessive pairing of genes. It means that we can trace this up to both of the parents. It means each of those parents would have had to have carried that recessive allele in order for them to pass it on and for it to be present in the offspring. So they would be heterozygous for the condition. And we use that principle to approach all of the individuals in our pedigree diagram. Okay, right. So you can see that, what I've just explained to you over there. One of the most common mistakes when it comes to pedigree diagrams is we confuse sex-linked to autosomal disorders. And we end up approaching our pedigree diagram as a sex-linked disorder when we are tracing it from generation to generation when it is not. So be sure to read your question carefully, and you can approach your pedigree diagram correctly. Okay. Many times here, looking at a pedigree diagram, at the end of a series of questions, we're going to have that final question asking us to do a genetic cross. So once we've worked out the genotypes, we'll have the phenotypes. Once we've worked out those genotypes, we're going to be asked to cross. And if we've incorrectly worked out our genotypes, it means our genetic cross is also wrong. So it's very, very important for us to spend a little bit of time on our pedigree diagram. Here are a couple of common questions. Explain why individual A, in this case, we're looking at albinism not being sex-linked, and why individual A has albinism. This would mean that that recessive pairing is a result of both parents. So here we approach it by explaining how the allele is passed on one from each parent. They have paired through fertilization in the offspring, and that is why that phenotypic trait is expressed in person A. Okay? Provide the genotype for individual B. This is the skill of taking what we know about those genotypes and reverse engineering them to the genotypes of the parents. And in this case, we're talking about parent B. Okay? And then point number 3, explain why the allele for albinism is a recessive allele. So now we look at the fact that, right, if person A received the allele from each of the parents and neither of the parent actually has the condition, it would mean they're heterozygous for the trait, and the dominant trait would be for normal pigmentation. And that's how we're going to explain the answer to question 3. Okay. Some of the most common sex-linked conditions, remember we've just discussed one that's not sex-linked, but some of them that are sex-linked include hemophilia, color blindness, and muscular dystrophy. So watch out for those in your questions. Okay. Evolution. Here we're looking at the majority of your paper, 54 marks, almost 1/3, slightly more than 1/3 of your paper. Okay. If we're looking at the definition for evolution, here we're looking at evolution as being the change in the characteristics of a species over time. Okay? Very simple explanation. Okay? And when we look at why evolution, why the theory of evolution? Because evolution is regarded as a theory because it is backed up. Many hypotheses have been proven, okay, over the years, which supports this scientific theory. And therefore, it has been verified. And that's why we're learning it. Okay, so some of the evidences that support evolution include these. Now, we did some of these already in grade 10, so you might need to go back to your 10 notes because it's a very long time ago and we don't always remember. Okay, but we do need to revise. So when we're looking at the fossil record or fossil evidence, we'll need to go back to our grade 10 work. Biogeography, we go back to our 10 work. Modification by descent and genetics is something you would have learned this year. Each of these add to the evidence, add to the value that we place on the hypothesis that has been generated, all backed up by these 4 points of evidence. Okay? If we're looking at variation within the same species, variation is determined by a number of things. Now, doing meiosis, we've already pinpointed a few areas of variation. And if you go back in the slides, once you have the time, you'll see that in prophase 1, the crossing over of bivalent chromosomes is what leads to variation in offspring. Not only that, but the random arrangement of chromosomes as well Okay, that also leads to variation. And you'll see here, I've included them in the points there for you. Point 1, during meiosis, we're looking at the random arrangements of chromosomes and the crossing over in prophase 1. Mutations to DNA also leads to variation on a much smaller scale, but this could create bigger problems for the organism. Some of these mutations could lead to beneficial changes. Other mutations, maybe not so beneficial, which could impact on survival at the end of the day. Random fertilization and random mating. Point number 3, random fertilization. Why that ovum? Why that sperm? Okay, there is a randomness to the fertilization. And then also, when it talks about random mating, why those two individuals partnering up to produce offspring? So there's a randomness to that as well. So we're looking at all of these. We need to make sure that when we look at our question, are we talking about variation due to meiosis? Because then we refer to the random arrangement of chromosomes and crossing over in prophase 1, or are we talking about variations within a species? Because if we're talking about species, we can include the other points as well. We can include the random fertilization and the random mating. All right, and here you can see that when we're looking at the different types of variation that occur within a species, we're particularly looking at continuous and discontinuous variation. Continuous variation, and this is where terminology becomes very important, it refers to a trait where there is a range of intermediate phenotypes. Each of those words is extremely important in getting you a mark for this definition. We're looking for the word range of intermediary or intermediate phenotypes. That is continuous variation. And an example here is height. We've got one end of the scale, we've got the other end of the scale, and there are many different intermediaries between the two. Discontinuous variation means we've got categories into which we can put the different versions of these traits. And an example here is blood groups, which we did in the genetic crosses. Okay? We do need to know the difference between these two, and we do need to know how to define them. The ideas of Lamarck, Darwin, and a concept of punctuated equilibrium in our understanding of evolution is very important. I've seen many final exam papers asking questions on each one of these. We generally focus on either Lamarck or Darwin, but punctuated equilibrium, in terms of a definition, is asked very often. Okay, so now we're looking at Lamarck quickly, and his theory was one of the first theories when it came to evolution. Okay, and he used two laws to motivate his theory. He looked at the law of use and disuse, and the law of the inheritance of acquired characteristics. Now, looking at these two laws, there's a little bit of a problem, because we've already studied meiosis, and we know that inheritance does not happen when we talk about acquired characteristics. Okay? And that's why his theory was not accepted widely in the scientific community. Okay? So when we refer to Lamarck, we'll have to apply his theory to a given context, but not only that, we're going to have to be able to identify with the fact that his theory actually has two laws. The first one is the law of use and disuse, and the second one, the law of acquired characteristics. Okay, so now we're applying the theory of Lamarck to why giraffes have long necks. Now, I know you've probably done this in class, but we need to put emphasis on certain things here. When we are talking about Lamarck and his theories, we need to make sure we reference that, in this case, looking at giraffes, all giraffes originally had short necks. Okay? And because they used their necks, they stretched their necks in search for food higher up in the trees, their necks stretched and became longer. I know, sounds a little bit strange, but bear with me. This acquired characteristic of having a long neck was then passed from generation to generation, and now that is why we have giraffes with long necks. Okay, let's point out quickly where his theory of use and disuse comes in. Firstly, his law of use, giraffes stretched their necks because they frequently used their necks to reach up into the trees for food. His law of the inheritance of acquired characteristics, this acquired trait was passed on from generation to generation. Okay, so this context we need to apply, or this answer we need to apply to the different contexts that we're going to be given in our final paper. All right, so study this point for point. It's generally 4 or 5 marks long. We have a possible 6 there. We just need to make sure that we write enough to get full marks for this question. Okay, and you'll see at the bottom, sometimes we just don't apply the formula. In other words, step 1 to step 6. So if you're a little bit unsure, learn the formula and apply it to the context of your question. The ideas of Darwin. Now, Darwin's theory was about natural selection, or evolution through natural selection. Okay? And here we're talking about variation within a population, but the passing on of only the favorable characteristics through the generations. These favorable characteristics, you'll know, are a result of mutations, okay, during meiosis or during DNA replication into gametes. But regardless of that, favorable characteristics are passed on through the generations, and this is going to help the populations of the species survive better. All right. Explaining evolution in a specific example, once again, the point where a lot of students make mistakes. Okay? So make sure that your understanding of Darwin's theory is applied to the context of the question that you're given. And if we look at the giraffes with the necks, like we did with Lamarck, let's see how Darwin would have explained this. Darwin would have explained it a bit differently. Okay? He was all about variation. So originally, there was a lot of variation within the giraffe population. And this is a continuous variation, isn't it? We've got long necks, we've got short necks. There was variation within the population of giraffe. Some had long necks, some had short necks, due to limited availability of food. So here we have the selective force, which is going to determine the characteristic that is favorable. And in this case, because of the availability of food being scarce, it was only the giraffes with the long necks that could actually reach the food. This became the favorable characteristic. Those with short necks couldn't reach the food and they died. Those that survived, and this is particularly those giraffes with long necks, were able to reproduce and pass on this trait to their offspring. And that is why we have giraffes with long necks today. Okay, so be sure to apply the formula that you see now to the context that you get given in your exam. Okay. Right, punctuated equilibrium. This is one that's maybe sometimes not as easy to understand. So if you're not too sure about it, just go and study the definition. So when we're looking at punctuated equilibrium, it basically takes Darwin's theory of evolution or gradualism over time, the changing within a species, and it adds to that. It says that, okay, well, here we're taking natural selection, and natural selection doesn't actually take place over a very long period of time. Natural selection takes place in punctuated bursts. Okay? And it's defined these points of rapid change within a species as the punctuation. And then the time period where a species does not change much as the equilibrium, no change. So when we're talking about punctuated equilibrium, we're talking about periods with little or no change, and then very short punctuated periods with rapid change through natural selection due to changing environmental pressures. That is what punctuated equilibrium is. Okay. Artificial and natural selection. We'll have to compare the two. Now we know a little bit about natural selection, and we know that nature is the selective force. Something in nature determines that favorable trait that will allow a species to survive. Okay? The selection is response to the suitability of the environment, and it will occur within one species. If we look at artificial selection, this is a selection based on a human's wants or needs. So humans become the selective force and not nature anymore. Selections to satisfy a need of a human being, and it can occur in more than one species. So when we are comparing these two, and that's generally the question you're going to get in your final exam, is either an explanation of the two or a table of comparison of artificial versus natural selection. So make sure you study this. It is quite important. And when we're drawing a table, it is a skill, so we make sure that our table always has a heading which includes both of the variables that are being compared. In this case, it's artificial and natural selection. Speciation. Every year there is a speciation question. The context, however, changes. Okay? And here you're going to see that speciation, due to geographic isolation, occurs in certain stages. Very important for us to know step by step how to approach this question. All right? Speciation means the formation of new species. And how does this happen? One of the biggest mistakes that are made by students here is that when we refer to speciation, we don't actually talk about populations of the same species, but we refer to species. Okay? And that gets a little bit confusing, and it's, depending on how you write it, it's scientifically incorrect. Okay? So we approach speciation step by step in the following way. If a population of a specific species becomes separated, think of a species living in a group. So in other words, one population of a given species is split down the middle. We now have two subpopulation groups of that once one larger group. Okay? They become separated for a number of reasons. The first reason could be sea, a lake, mountain, river. There will be no gene flow between these. They will not be able to interbreed, and each population will be exposed to environmental pressures, selection pressures, and therefore each will become genotypically and phenotypically different from one another. And even if they were to mix again, they would not be able to interbreed. Okay? Right, here we don't confuse population with species. All right, moving on. Reproductive isolation mechanism. So basically this is, why do species breed with one another and why do other species not breed with species to come? So these could include, there are 5 points for us to go through. We would simply have to be able to identify these, but be sure to take the slides, print them out, and then you can annotate them a little bit more with your notes that you've taken from class. Reproductive isolating mechanisms. The wording of these is very, very important. Moving on to human evolution. This is the last section that we're going to be doing. Firstly, if we look to classify human beings, we find that through the kingdom of Animalia, we can break it down to a point where we have the family, the genus, and the species. In many papers, it asks, what family do humans belong to? And you'll see the family here is Hominidae. Not hominins, not hominids. The family is Hominidae. We need to be sure of how we write that to get the mark. Okay. Similarities between humans and African apes. I'm not going to spend a lot of time on this. This is also dealt with in the Junior Tuckies online platform. Large brains, skulls compared to body mass. These are the similarities. Okay? Make sure you study these in sequence. It's asked quite often, along with the differences. Now, there's a lot of information that can go in between here. Okay? But the biggest problem when answering questions like this is that when we're comparing the differences between humans and African apes, is that we don't read the question properly. If it asks the differences between the skulls of African apes and humans, we cannot include all of these points. We need to select those that are particular to the skull and only the skull. Okay. We're looking at evidence that supports the common ancestor of living hominids. The fact that fossil evidence shows we were all most likely bipedal. There is a prognathism, the dentition, the cranial size, and the fact that there are opposable thumbs. Genetic evidence supports the common ancestor through mitochondrial DNA, and cultural evidence where we trace the use of tools through history. Okay, here you're going to see a phylogenetic tree, and understanding a phylogenetic tree is important for us because we will need to be able to pull some information off this based on the questions that we asked. So firstly, we can see that these are the two species that are alive today. Based on this phylogenetic tree, we can see present days at the top, and the furthest point in history is down at the bottom. We can see that the species or the genus Homo habilis, the genus Homo, species habilis, existed at that point and went extinct at about that point in time. So reading off of the phylogenetic tree is important for us. And we can see that at that point on our phylogenetic tree, both Homo sapiens and Homo neanderthalensis were both around at the same point. Okay, so they coexisted. Okay, evolution, human evolution, 54 marks again. Here we're going to be looking at the specific fossils. We'll need to be able to study, or we'll need to know Ardipithecus. Very important, and I've jotted down a few points to remember, to study. We'll need to know Australopithecus. Okay, we'll need to know the time frames, the fossils that were found only in Africa, and Australopithecus africanus, the tongue child, the Mrs. Pless. These three fossils you'll be asked about, and you'll be asked with the names Lucy, tongue child, and Mrs. Pless. Some important points for us to take note of there. Human evolution. We're going to look at the Australopithecus sediba. I've given this its own slide because it's considered a transition fossil. What does this mean? That means that when looking at Australopithecus sediba, we find that it has both characteristics of the Homo genus as well as Australopithecus. So we find this as being that transition species, and they like to ask that. They want to know your understanding of the word transition species. It has the characteristics of both the Australopithecus as well as the Homo genus. All right. Homo habilis, also very important. This is the handyman. This is the point in time where tool use became very, very prominent within the evolutionary chain of hominins. 2.5 to 1.6 million years ago. Fossils only found in Africa. Okay. Homo erectus. Now, this is a point in time that's very, very important. If we're looking at Homo erectus, the oldest fossils of Homo erectus were only found in Africa. Some of the younger fossils were found in Europe as well as Asia, which means at some point, Homo erectus left Africa. That's evidence to support what we were going to be looking at in 1 or 2 slides' time, the out of Africa hypothesis. Okay? Oldest fossils found in Africa, younger fossils found in other parts of the world. Used simple tools, but you can go through some of the finer details there in your study. Right, this is the out of Africa hypothesis. 4 points, and it is asked every year. It's when the first hominins left Africa. Here we're talking about Homo erectus in the first event of the out of Africa. This happened 1.9 million years ago. This is a very long time ago, in the first event. So they populated both Europe and Asia. If we're looking at event 2, about 70,000 years ago, now we're not talking million years ago, we're talking 70,000 years ago. Okay? A group of Homo sapiens also left Africa. And what happened is they populated Europe and Asia as well, and they outcompeted those early species of Homo erectus for resources, which means the Homo sapien became the surviving species in those parts of the world. There are a couple of points for us to go through. Remember, we're looking at 54 marks, human evolution, within paper 2, so we need to place a lot of emphasis on this, and that's where I would actually start. It was the most recent in our preparations, ending off in term 3, so that's where I would start with my preparations. So I hope this is going to help you, and please make sure to visit the Junior Tucky online platform for more very, very important resources in preparing for your final exams. Thank you very much.